Namaskar Mitranno. Welcome to today's explainer. I am absolutely thrilled you're tuning in because today we're tackling one of the wildest challenges in the history of science. We are talking about the ultimate biological sorting problem. Think about it. How on earth did scientists look at every single living thing on our planet and actually figure out how to organize it all? Let me tell you, it took some serious scientific jugaad, a whole lot of trial and error, and honestly, some absolute genius to figure it out. Okay, let's dive right in.
87 million. According to the 2011 census, there are a staggering 87 million species of living organisms just hanging out on Earth. And that includes both land and sea. I mean, just imagine you're hosting a massive big fat Indian wedding where absolutely no one knows their seat, and you are the one who has to organize the seating chart. Now take that chaos and multiply it by 87 million. Every single species has adapted differently based on its geography, what it eats, how it defends itself. We even see crazy differences between organisms of the exact same species. It is literally total biological chaos.
So the million-dollar question is, kaise divide karna? How do we possibly divide 87 million distinct species into groups? To even begin studying such a vast number, creating groups and subgroups based on their similarities and differences was ekdam essential. This whole massive undertaking of dividing living organisms into manageable groups is what we call biological classification. But as you can probably guess, getting it right definitely didn't happen overnight.
Section 1. Early attempts at classification. You know, before we got a reliable system, scientists really struggled to sort out this incredible mess. They tried, they failed, they revised. In 1735, Carl Linnaeus started super simple. He divided the living world into just two kingdoms, vegetabilia and animalia. Basically, just plants and animals. But as microscopes got better, well, that just wasn't cutting it anymore. So in 1866, Haeckel proposed three kingdoms, protista, plants, and animals. Then, jumping to 1925, Chatton created a totally different two-group system based on cell types, prokaryotes and eukaryotes. Still, the picture wasn't quite complete. By 1938, Copeland stepped up, dividing living organisms into four kingdoms, Monera, protista, plants, and animals. It was definitely getting better, but the suspense was really building for a truly comprehensive system.
Section 2. Enter Robert Whittaker and his 1969 master plan.
That brings us to our hero of the day. Enter Robert Harding Whittaker, a brilliant American ecologist. In 1969, he stepped in and completely changed the game by dividing living organisms into 5 distinct groups. Chala, let's understand this master plan together. Just look at how beautifully and logically this chart organizes all life. You can clearly see the main split between prokaryotes and eukaryotes right at the top, which then filters down into unicellular and multicellular, ultimately giving us Kingdom 1, Monera, Kingdom 2, Protista, Kingdom 3, Fungi, Kingdom 4, Plantae, and Kingdom 5, Animalia. It's a total masterpiece of organization. But the real question is, how exactly did he decide who goes where?
Well, Whittaker didn't just guess. He was like that strict but super fair tuition teacher we all had. He used 5 exact criteria to make his classifications. First up, the complexity of cell structure. Second, the complexity of the organisms themselves, like are they unicellular or multicellular. Third, their mode of nutrition. Are they autotrophic, like plants, saprophytic, like fungi, or heterotrophic, like animals? Fourth, their lifestyle. Are they producers, consumers, or decomposers? And finally, fifth, their phylogenetic relationship, which just means how they evolved from simple to complex forms over time.
Let's quickly simplify that first and honestly most important rule, cell structure. I want you to think of a prokaryotic cell like a very basic one-room studio apartment. Everything is just out in the open. These are cells without a distinct nucleus or cell organelles. On the flip side, eukaryotic cells are like a proper multi-room flat. They have closed doors and specific rooms for specific things, meaning they have a well-defined nucleus enclosed right inside a membrane. Keep this housing analogy in your head. It's going to be very helpful as we go on.
Section 3. Kingdom Monera, the solo workers.
I actually really like calling Monerans the solo workers of the microbial world. Let's look at a fantastic contrast right here. If you were to take a tiny drop of curd or buttermilk, dahi or tak, put it on a clean glass slide, dilute it, and look under a high-power microscope, you'd see these moving, small, rod-like microbes. Those are lactobacillus bacteria. Now compare that to the irregular, shape-shifting microbes like amoeba on the other side, which actually belong to our next group, Protista.
They look totally different from each other, right? So, what exactly makes a moneron a moneron? Well, all types of bacteria and blue-green algae are included right here. The key traits are actually quite simple. All organisms in this kingdom are strictly unicellular. They might be autotrophic, making their own food, or heterotrophic, relying on others. But the absolute most important thing, they are prokaryotic cells. Remember our real estate analogy? They are the ultimate studio apartment solo dwellers. Absolutely no distinct nucleus or cell organelles whatsoever. Samajh lo? Simple, independent, and very basic.
Section 4. Kingdom Protista, the motiles.
All right, now let's step it up a notch with organisms that have a bit of jugaad built right in to help them survive and move around. Welcome to Kingdom Protista. If you prepare a temporary mount of just a single drop of pond water on a slide and peek under the microscope, you'll find these incredible motile microbes just darting all over the place. You've got the spherical volvox, the slipper-shaped paramecium, the euglena, and of course the famously irregular amoeba. So, the crucial point to remember about Protista is that while they are also single-celled organisms, just like Monera, they are eukaryotic. They've officially upgraded to the multi-room flat. They possess a well-defined nucleus, nicely enclosed in a nuclear membrane. Plus, they have these amazing built-in transport systems for locomotion, things like pseudopodia, hair-like cilia, or whip-like flagella. Some of them, like euglena and volvox, contain chloroplasts and are autotrophs. Others, like amoeba and paramecidium, are heterotrophs. They are much, much more complex than our studio-dwelling monerans. Barbar?
Section 5. Kingdom Fungi, the recyclers. Okay, be honest. Have you ever taken a moist piece of bakri or bread, kept it in a closed dubba, and then completely forgotten about it for a few days? After 2 or 3 days, this fine cotton thread-like tuft starts growing all over the surface. That, my friends, is fungi in action. Now, to truly understand fungi, you absolutely must understand this specific word, saprotrophic. This means they are organisms that feed upon decaying organic matter. Think of them exactly like nature's very own compost bin. They break down dead material, absorbing their nutrition directly from decaying organisms. Look, it's a dirty job, but someone has got to do it for the Earth's ecosystem to keep functioning.
What's really fascinating here is the sheer variety of them. When we prepare a temporary mount of those bread threads under a microscope, or honestly when we just look around in nature, we see fungi absolutely everywhere. Great examples include baker's yeast, aspergillus, which is a fungus that loves to grow on corn, penicillin, and even the everyday mushrooms you might order on a pizza. They might look a bit like plants, just sitting there quietly in the soil, but trust me, their lifestyle is entirely different.
Let's lock in their main characteristics. Fungi are strictly non-green, eukaryotic, heterotrophic organisms. Because they are non-green, they absolutely cannot perform photosynthesis like plants do. No sunlight snacks for them. They do have a cell wall, yes, but it's not like a plant's cell wall at all. Theirs is made up of an incredibly tough and complex sugar called chitin. Also, many of them are thread-like, and you'll actually find multiple nuclei present right there in their cytoplasm. They are highly specialized, brilliant little recyclers.
Now, even though several new systems of classification were introduced after Whitaker's in 1969, his five-kingdom system remains incredibly popular and widely accepted today. It brought real order to the mind-bending chaos of 87 million species by logically grouping them, from the simple studio apartments of Monera all the way up to the complex multi-room setups of eukaryotes. But science is all about constantly questioning, right? No system is absolutely perfect. So I'm going to leave you with this. Based on everything we've explored in this explainer today, what do you think are the merits and demerits of Whitaker's classification? Where might it actually fall short? Bichar kara. Use your brain power, think critically about it, and keep that scientific curiosity alive. Thank you so much for joining me today, and keep exploring this amazing living world of ours.